So is everybody registered to vote?
Yes.
Yes. Deadline is tomorrow. So if you don't vote, then you can't complain about what politicians do. So first thing. Second is guest lecture on Wednesday on flaviviruses. Somebody who knows a heck of a lot more about it than me from OHSU. And I'm not sure if he's going to be recording a lecture. So if people want to do that, that's a warning now to go ahead and do so. This one is recording, at least I think it should be. The other thing I'm not sure if he's going to do, is clicker questions. So, but of course I will. So we can start out with those. Unlike most of the other virus capsid proteins which we've discussed so far, the Tobacco Mosaic Virus, wait, that was last week, capsid protein has a beta barrel structure, double beta barrel structure, alpha helical bundle structure, coiled-coil structure, cross-linked-capsid protein. Once I had somebody who turned in a Scantron and everything was seen.
Did they pass?
No. Ten. Okay, the results are on the wrong screen. Voila! Yes, even if you didn't remember, if you go back and look at your notes, that TMV does have this alpha helical structure all wrapped together as a bundle. We've kind of been beating on the whole beta barrel structure, I think, a whole bunch. So be unlike all the other ones. So you can kind of eliminate at least the first two to start with. And then what about the coiled-coil structure? Where have we seen that? Coiled-coil structure. Coiled-coils are alpha helices wrapping around each other. The H protein in 5X174 that makes this amazing sort of barrel with coiled coils. What about cross-linked-capsid protein structures? Bacteriophage, HK97, etc. So, do we have any more questions on plant virus stuff before we move away and talk about, boring animal viruses? No? Yes? Unhappy? Happy? What's the most amazing thing about these viruses?
They can confer benefits.
Well, they can confer benefits. Yeah, thermal tolerance is definitely one of them. But at least as far as I'm concerned, one of the most amazing is that you've got these multiple genomes in multiple capsids, which is just totally bizarre, at least as far as I'm concerned and from the virology point of view. On the other hand, the picornavirus viruses, I'm not sure how this happened, but most of the people who work on these things call them picornaviruses. Why? They're small RNA viruses, and I think they just wanted to have a cool-sounding name. So most of the time when people talk about them, they'll call them picornaviruses. I will call them picoRNA viruses because I think it's a lot easier to understand because they're small RNA viruses. Mostly what people talk about when they talk about picoRNA viruses is polio, and we'll spend a couple of minutes talking about polio and particularly polio eradication. We're getting really, really close. Hopefully in the next, I don't know, probably 5 years, we'll have eliminated wild polio virus, but then there's some other issues with non-wild polio virus and how we eradicated it in the first place. And we'll talk a lot more about that as we move through. Polio, of course, is known as being a neurotrophic virus, causes neurologic disease, but almost all of these picoRNA viruses have nothing to do with neurologic, and they're mostly enteroviruses. What does enterovirus mean? In your belly, mostly, intestinal tract. And that's also where polio is for the most part, which is partly why it's a bit of a problem to try and get rid of. So that's the disease aspect of things. The important stuff, all the molecular biology, has to do with entry and replication and translation of these particular RNA viruses. And again, replication is something which is clearly very viral because it's RNA-dependent RNA polymerases. And translation, which is what irises are all about, is clearly an issue, particularly if we're talking about replication versus translation, and we've already talked about this for a number of different viruses. So our classic outline, what are the important things that Stedman really cares about? Replication and translation down here. Again, we'll talk a little about the origin. We've already talked a bunch about the structure. What kind of structures the capsid protein have? The capsid protein is beta barrel, so like most of the ones we're talking about. Binding and entry, it turns out binding and entry for most of these picoRNA viruses, it's a lot more like bacteriophage than it is like your sort of classic animal viruses, membrane fusion, endocytosis, etc. Look a little bit at the genome. The genome here is really quite interesting and very different from the RNA viruses of the bacteriophage that we've talked about so far. It's made as a polyprotein, and we haven't really talked too much about polyproteins, but this is one big, ginormous open reading frame that then gets chopped up into smaller pieces in order to make the actual virion and also make all the enzymes that are required for that function. So it's an interesting process, but as we'll see, really common for a lot of the RNA viruses that are infecting animals. Then, again, replication, translation. Replication here, RNA-dependent RNA polymerases and how you get that replication taking place. Translation has to do with irises. Anybody remember what an iris is?
Internal ribosome entry.
Exactly, internal ribosome entry site. And so basically the name says everything. It doesn't start at the 5' end. It starts somewhere in the middle. And then how these guys get out, we'll talk about very briefly at the end. So picoRNA viruses, the vast majority of what we know, not surprisingly, has to do with the one which causes the most nasty diseases, and that's poliovirus. And these are some members of the enteroviruses. There's also Coxsackie virus, echovirus, and then a few more enteroviruses that we'll talk about at the end. Enterovirus 68. Anybody heard about it? No? It's amazing how quickly people forget. 2 or 3 years ago, there were like 1,600 cases in California and a couple of cases of infant paralysis. But, gee, how quickly stuff disappears here. There also are a number of other viruses which are, in fact, not in the enterovirus group. Rhinoviruses. Rhino means what? Nose. What are these causing? Colds. And in fact, these are probably the most common animal viruses out there. We're probably infected by multiple ones most of the time. Turns out that they are very similar in terms of how they replicate at a cellular and molecular level to a lot of these enteroviruses, but of course, they're replicating not in the gastrointestinal tract, but in noses and what's connected to them. A few other viruses, cardioviruses, aptoviruses, etc. Just also wanted to mention the hepatitis A virus down here. We'll talk about hepatitis A, B, and C in this course. It's rather confusing because they're completely different viruses in terms of how they replicate, but they cause similar kinds of disease, and so that's why they're named as such. So keeping track of these is sometimes a bit challenging. A is a picoRNA virus. B is a really funky retro-like virus. And C, as we'll talk about, well, hopefully next time our guest lecture will talk about it, is one of the flaviviruses. So they also here in the table in the textbook, 4 additional picovirus genera that are not listed here. There's probably a whole bunch more that are out there. In fact, a lot of those little spots that you see or that we saw way back when, lecture 1, little spots of nucleic acids in seawater stains or earth or whatever, a lot of those turn out to be picornavirus viruses as well. There are very, very large numbers of them out in the environment. So, but again, we're mostly going to concentrate on poliovirus because pretty obvious reasons having to do with disease. Generally, a poliovirus infection is not problematic, and this is one of the big problems of actually eliminating polio, is that it's only about 1 in 200 people who are infected with polio who actually have then neurotrophic disease. And that means that there are lots of people, particularly when you're vaccinating, that you're never going to know whether they've had polio or not. And so detection turns out to be a really big problem. However, when you do have paralysis, and this is particularly true for kids, is it can be pretty deadly. 5 to 10% of those who do get paralysis, which is about 1 in 200, 0.5%, end up their lungs get paralyzed, some other kind of major internal muscles end up being paralyzed, and it's really a problem. And also, this is really a big problem for kids. There is a vaccine strain, in fact, the major vaccine strain that's used worldwide, not in the US, and we'll get back to that a little bit later on, is the Sabin strain, which is an attenuated vaccine, and attenuated just means it replicates, it makes more virus, but it's not neurotrophic. It doesn't go into neurons most of the time. And interestingly enough, the change in this particular virus is in the iris sequence. So in this sequence, it's really important for translation. We'll get back to a little bit later on. And so again, this is the major strain here, which basically is replicating in the gastrointestinal tract. It's perfectly fine. Doesn't seem to cause paralysis, at least when you don't have a reversion in this iris sequence that we'll get back to. So we've known about poliovirus, and we think we've known about poliovirus for over 5,000 years, or at least the symptoms thereof. Here's an example of a 5,000-year-old carving stolen from a tomb in Egypt, and now I think in Copenhagen, of a guy with acute flaccid paralysis in one leg that looks really strikingly similar to this much more modern patient right here. And this is really typical, particularly in legs. Also, you'll see them in arms of kids. Before there were vaccines, there were over 35,000 a year cases of polio in the US. And the big advance in terms of actually being able to make vaccines was being able to grow viruses in cell culture. So not actually having to use animals to produce the virus. And then the original vaccine was an inactivated virus. So make large amounts of active and pathogenic virus and just kill it. Now, kill it's got to be alive, right? In terms of viruses. Or inactivated. And this was the Salk vaccine. And interestingly enough, that was 51 years ago that it was developed. There was a big party last year about 50 years of the development of the Salk vaccine. And what was really pretty amazing is within 2 years of that being announced, 90% reduction of polio in the US. And now completely gone by 79, at least the wild type strains. We'll get back to that a little bit later on. I mentioned the attenuated vaccine. Attenuated is really good, particularly in the developing world, because it actually does replicate. And one of the great things about an attenuated vaccine is you give it to someone, and again, they have no phenotype, but it's replicating. And that particular virus will actually be shed. It actually will be coming out of the gastrointestinal tract in the normal way. And then it can infect other people who actually haven't been specifically immunized. And so you have this sort of circle effect of immunization that happens when you have an attenuated vaccine. And that's probably mostly why this attenuated vaccine has really been the one which has been used throughout most of the developing world. The other issue with the inactivated vaccine is that you need needles. You need someone much more trained in terms of vaccination in order to administer it. And we'll talk about the problems with the attenuated version, which I've kind of mentioned already. as we move along here. So pretty much taken care of in the U.S. Why do we care? Oh, sorry.
Oh, quick question. How is it inactivated? Is that heat or pH or specifically this?
Oh, the inactivation of the Salk vaccine is formaldehyde, so it's a chemical inactivation, which they use. And unfortunately, some of the first batches they didn't completely inactivate, but that's another story for another time. This was, again, completely eliminated in the U.S. Before 1980. So, great stuff, no problem, right? So we can stop worrying about it. Well, except for the fact that we're all connected to the rest of the world. And there were, again, in the 50s, basically right as this was happening, about 500,000 deaths a year from polio worldwide. And even in 1988, so a good 10 years after it was eliminated in the U.S., still about 350,000 people dying worldwide, again, mostly in the developing world at the time. And that's when the WHO and a bunch of other people said, okay, we need to try and get rid of polio. And what's pretty amazing is now, last year, there were 74 cases. So from 350,000 to 74 is pretty darn amazing. There's been a pretty large investment that's gone into that. But the estimates are that if we can completely eliminate polio, it'll save on the order of $20 billion a year. So I think it's a pretty darn good investment. And I just wanted to skip through a couple of next slides pretty quickly, looking at the successes of some of these things. And literally in the last 5, I guess this is now 6 years, this is what poliovirus, wild poliovirus looked like 2009, 2010. Very widespread in West Africa, Central Africa, India, Afghanistan, and Pakistan. In 2012, much less of this. Few cases in Nigeria and Chad, India, Pakistan, and Afghanistan. 2014, a couple more cases are popping back up here. One thing that I didn't mention before, but there are 3 main strains of polio, at least the ones that are causing this kind of paralysis, 1, 2, and 3. 2 and 3 have been pretty much completely eliminated by 2014, and actually 2012 was the last case of poliovirus type 3. Still type 1 circulating, and now, as of this year, we're down to just 2 countries in the world, which unfortunately, as we know, are not terribly politically stable right now. And then there are a couple of other things that I wanted to mention. You may notice these purple dots over here in also rather remote regions of Thailand and Laos. These are what are called VDPV. What the heck is that? Vaccine-derived poliovirus. So this is reversions of the mutations in that iris sequence, which are now causing poliovirus disease. And so that is becoming a larger and larger proportion of the actual poliovirus. And this is when I say poliovirus, these are cases of paralysis that are happening through poliovirus, not just the 199 out of 200 that you have no symptoms for. Those are really hard to detect these, particularly in places like the tribal regions in Pakistan, etc. So, and this is literally from beginning of this year back up to last week of these reported cases, 11 wild-type polio and 3 of these vaccine-derived polio. Look at the real success story, as far as I'm concerned, is Nigeria, Central Africa, one of the most populous countries in Africa. There were also quite a few cases, mostly up here in northern Nigeria, which, if any of you know your global geopolitics, is also a not terribly stable area. Boko Haram is also highly active in this part. 53 cases in 2013, 6 in 2014, and none in 2015, which is a real tribute to the local public health groups there and really being able to completely eliminate polio. And one of the things that gets a little bit hard to see here, but the red dot is wild polio, and all these green dots are vaccine-derived. So should also give you an indication of what we need to deal with. Our problem children as far as polio eradication are concerned are Afghanistan and Pakistan. But even there, we're making a huge amounts of progress despite the political unrest in both of those places. Afghanistan in 2014 had 20 cases. These are the cases in 2015, only 5 of them, which is, again, really pretty impressive. And Afghanistan is probably going to be declared polio-free in the next year or so. Now, there are some issues with that. And the major one has to do with reporting. How do you know if there are polio cases there? And are they being appropriately reported? And there's a whole set, and I'll give you some of the links if you want to take a look at this a bit later on. But basically, it has to be some kind of acute flaccid paralysis. So again, the same kind of thing you see with arms or legs. But then also has to be confirmed as polio with one of the various different strains that's associated with it. And so you also have to collect stool samples, have to be multiple positives, et cetera, towards doing that, which is hard when you're in the middle of a war to do some of these things. Afghanistan, again, is doing really amazingly well as far as this is concerned. Pakistan has a bit more of a problem, and this is also in the so-called tribal areas of Pakistan, very close to the Afghan border, where there's a lot of activity of multiple different, and this is blowing this up, this is from last year. Here, almost all of the poliovirus activity are in these, again, tribal areas, relatively lawless areas, but also in some of the major conurbations. Anyone know what that dot is? That's Karachi. And so there's lots and lots of movement going back and forth there. So it's getting close. We're basically narrowed down to more or less one country. Again, I think in Afghanistan it's probably going to be gone in the next year or so. Dealing with Pakistan it's getting a lot better. I was actually really concerned about 2 or 3 years ago this is ever going to happen, but it really seems to be making progress. Yeah.
In a case of like Nigeria, where you have a whole year with no new cases, can you breathe a big sigh of relief already, or do you need to wait 5 years so that there isn't something lurking? Or
well, the thing I think with wild poliovirus, it's really pretty good in Nigeria right now. I think the issue is going to be in the vaccine-derived, where there's still going to be some of that. And one of the things that's happening now is what people are talking about, polio eradication. And so there's a great website that gets into this in all kinds of good detail about how to deal with those residual cases. Because as soon as you do have one of these cases that comes up, then you've got to make sure that everyone else is protected from it. And so that's, I think, is going to be the major issue. And the problem with what's happening now is most of the cases in places like Nigeria are all these vaccine-derived. They're not the wild version. This is the Sabin vaccine, which is great because when you immunize people, they'll immunize people around them. But there's still going to be a relatively low frequency. It's about 1 in 2 million cases where you have this reversion, but we're talking about billions of people on the planet. So 1 in a couple of million is a problem. And that's, in fact, what happened in the U.S. So in the U.S., there were a few people who came down with vaccine-derived polio, and based on that, they stopped using the polio drops, the live attenuated vaccine, and went back to using injected polio vaccine because that can't revert. It's inactivated. It's dead. But it's a lot harder and way more expensive to use that kind of immunization. But that's the current plan, is to get rid of the oral polio vaccine and then switch over to the inactivated polio vaccine. That's the IPV here. In order to hopefully get rid of all of everything that's left. And so the idea is when you do have outbreaks like this, or in fact more like this, is you then have a massive influx of the injected inactivated polio vaccine to try and control any of those outbreaks as rapidly as you possibly can. And so that's the idea. Again, this is the way that it was being used as your oral polio vaccine. And here's the smiling baby getting a shot. Yeah, right. And you have kids? No, that's not the way it usually works. So on that note, we'll have another clicker question, and this should hopefully be very straightforward. In the next few decades, the most likely source of polio is type 2 wild virus, type 2 vaccine-derived, Nigeria, India, or Israel.
Of course it's Israel. It's right there in the protocols design on.
Israel, I'd say, I can mention this now, but of course probably to tell you something about the clicker question. There was a big outbreak of vaccine-derived polio in Israel 3 years ago. So hopefully we can get 100% on this one. We'll see. I mention it, yeah. You better. It's lighting up, doing all its good things. Does anyone else want to vote? No? Vote early, vote often. What's the answer? Well, we'll see what you guys think the answer is. Momentarily. In 1 second. Two of you didn't like. Somebody's messing with me, exactly. I'm sure that's what's going on here. And somebody realizes that the number of clicker questions versus the actual number of points at the end of the class you're going to get are so small, it really doesn't matter.
Has CDER already happened?
So yes, it is vaccine-derived polio, which is by far and away the most likely to be what we'll be seeing in the next few years. So that's it for polio-derived disease. Oh, no, except David has a question.
So in the 1 in 2 million cases where it reverts and gives
Yeah.
you polio, but then this is the replicating virus, so then that version that's spreading from that patient is infectious for anyone?
Yeah, so the question here is, if you have a reversion in that virus sequence that we'll talk a little bit more about in just a second here, if you have a reversion that goes back to wild type, that's then infectious. And the key there is that, yes, that's infectious, but you have to be, if everyone around that person is immunized, then you're okay, it's not going to spread. But the problem is, if you stop immunizing, how do you know how long it's going to be until you have one of these, and they're all still replicating in the environment. So how long is it before you have some of these things? So there's a lot of questions about how long you have to keep immunizing for and when you can stop immunizing. Now, there hasn't been polio in the U.S. since '79. Are we still immunizing? Yes. So, yeah, these are some interesting questions that we really don't have very good answers to at this point. Okay, other questions on disease? Let's see. Molecular stuff, however. Now, this is the cool stuff. So we already talked a little bit about the structure of poliovirus when we talked about wonderful icosahedral symmetries. This is our pseudo T equals 3. What does pseudo T equals 3 mean? Multiple different proteins arranged in a T equals 3 form. And hopefully it's really visible here. Blue is our pentamer. Red and green is our hexamer. So we would go 1, 1, right? Easy, no problem. Give you something like this in the next exam, even though it's not structural. So the T3 proteins are, again, incredibly creatively named VP1, 2, and 3. And they're 60 each of them. So as usual with a, T equals 3 structure, you're going to have 180 in the actual capsid. Binding of the receptor for this virus, and we'll look at the receptor in just a second, happens in what are called canyons. Where the heck are the canyons? Canyons are when you look at this structure here, we have these big depressions. In fact, literally right around the 5-fold axis of symmetry, you see this big hole right here. And so normally when you think about a virus receptor, the classic case would be something like adenovirus, projections, the 5-fold axis. That's what's going to interact with your receptor. Here, the receptor is actually interacting with something which is pretty deep inside the surface of the poliovirus. And we'll see what happens as far as that's concerned. Turns out that one of the best not only poliovirus, because that's not so much of an issue in terms of antivirals, but anti-rhinoviruses, and it turns out rhinoviruses are very similar to this, is interacting with where that receptor binding would be right here. And so these are those holes that you have in the outside of the structure. Look at that in a really high resolution detail. Here you can actually see the canyons a little bit better. This is again the figure we looked at before. Here are these black holes right around the 5-fold axis of symmetry. VP1, VP2, and VP3 are very similar to each other structurally, and they all have this beta barrel structure here with all these beta strands wrapped around sort of a central piece. And this is supposed to show that they're basically trapezoidal, each of them. And so you can see the 5 of them coming together at the 5-fold axis, and then 6 of them coming together here at these 3-fold axes of symmetry. And this is a reminder, these are made as a polyprotein, which we'll look at in just a second. So, receptor binding, yeah, Peter.
Are these VP1 through VP3 the same ones we met elsewhere, or are they unique to this particular virion?
So the question is here, well, I'll paraphrase, are VP1s, 2, and 3s created equal? And the answer, of course, is no. This is poliovirus VP1, 2, and 3. And the rhinovirus VP1 and 2 and 3 are going to be pretty similar. You talk about our favorite virus in the Stedman lab, the SSV virus. We have a VP1, 2, and 3, totally different. You talk about other viruses, they'll have CPs as for their capsid proteins. Yeah, unfortunately, they're all going to be different in terms of their nomenclature. So this would be poliovirus, VP1, VP2, and VP3. So I mentioned already that we have binding here in the canyon around the 5-fold axis of symmetry. This is where the virus interacts with this receptor. That's shown here. Here's the poliovirus receptor, this orange thing right here, interacting again right around the 5-fold axis in this depression right here. So you have this interaction with the poliovirus receptor next to the cell membrane, and then you have a big conformational change, and that big conformational change has to do with VP4. Remember, VP4 is not one of the ones that are in the pseudo T=3 capsid on the outside. It's sitting on the inside until you have binding by the receptor protein. And then you have a big conformational change that happens, which opens a hole, which projects through the membrane, that should look, again, really, really familiar based on the bacteriophage we've been talking about. And then the genomic RNA is basically pushed through the membrane in this process. This genomic RNA also has a protein, that's what's in gray here, which is attached to the end. It's virus protein G, VPG, and we'll look at this in a lot more detail as we move forward here, but that is basically seems to be how this is getting transported. Is you have interaction with the receptor, conformational change of VP4 and part of VP1, which pulls this protein into the cytoplasm. That protein is covalently linked to the genome, so that genome comes inside the cell. So again, very, very similar to what's happening with bacteriophage on a idea of making a hole in the membrane and the genome getting poked through it. Receptors for poliovirus is very well known, and in fact not at all unlike the case for lambda, where the receptor was named based on the virus rather than what it normally did. Here also, the poliovirus receptor is named the poliovirus receptor, even though it's probably involved in cell-cell signaling in the absence of poliovirus. One of the things that I did want to mention here is that this receptor, which is very similar to a number of other receptors, all belong to the immunoglobulin superfamily. Why do you think immunoglobulins are being used as receptors by a whole bunch of viruses? Now, what do immunoglobulins usually do? Bind to things. So it's not surprising the virus takes advantage of something that's already been evolved to bind to things. And you'll see that many of these other viruses as well, picoRNA viruses, and we'll talk about many more other ones. Rhinoviruses also bind to a immunoglobulin type cephalomyocarditis virus, et cetera, et cetera, et cetera. Not all of them do. Some of these bind to slightly different ones. But for the most part, these guys are binding to immunoglobulin superfamily proteins. Again, just because they bind to things, and bind to things really quite specifically. What's that genome that gets brought inside the cell? We'll walk through this from 5' end to 3' end, because that's the way everything works, right? So the 5' end of the poliovirus genome is this VPG, so VP virus protein G. That's just part of the process. We'll talk about where that virus protein G comes from a little bit later on, but it's a covalently bound protein at the 5' end. You have a non-coding region. This often will be called in a messenger RNA what? A UTR. What's the UTR stand for? Untranslated region. So here there's non-coding regions, also non-translated regions. So here, we'll get back to a little bit later on, this is where the iris is. And then you have one really big open reading frame. This is about 1,000 amino acids in length here. And just literally one open reading frame, one translational start site, one stop codon. And then a untranslated region, and in the case of poliovirus, a poly A tail. And it turns out this poly A tail is actually encoded for in the genome. And so you always have this poly A tail, and it's always going to be there. And we'll see what that means in terms of how this replicates a little bit later on. So in this polyprotein, and polyprotein should actually be telling you something about what happens here, lots of different proteins that are going to be translated as one protein, but then chopped up into a whole bunch of other proteins. These ones should sound familiar. VP4, that's on the inside, VP1, 2, and 3. These are all the proteins which are making your capsid. These are all of the proteins that are not involved in your capsid, and not surprisingly, if they're not involved in the capsid, what are they going to be involved in? Making more of the genome. And particularly this protein right here, the 3D protein, this is the RNA-dependent RNA polymerase that we'll get back to a little bit later on. So if you have this VPG at the 5' end of your genome, does that look like a normal messenger RNA? No. So somehow you've got to get translation taking place. So what happens with a normal messenger RNA? We have a nice cap protein, cap binding proteins, EIF, 4E, 4G, et cetera, all of these guys bind to the cap structure, help the small subunit of the ribosome to bind together with eukaryotic initiation factor 2 and GTP, which then gets you your functioning ribosome. Okay, we don't have a cap anymore. Now what do? Well, now, of course, we have a iris, an internal ribosome entry site. Now, these internal ribosome entry sites, we'll look at these in a little bit more detail in just a second. They're lots of secondary structure here in the middle of an RNA. This is clearly what's happening in terms of the 5' non-translated region in poliovirus and most of these picoRNA virus structures. And basically what this structure does is it allows binding by the small subunit of the ribosome together with EIF2. It needs some of these other initiation factors, but most importantly, it doesn't need the cap binding complex and this bridging protein, EIF4G. And you'll notice little scissors up here. What do these scissors stand for? Not an endonuclease, that's close. But an endo, what else is around? We got nucleases and proteases. So it's a protease. So this is the protease. Turns out it's exactly the same protease that's used to chop up the individual proteins out of your massive original protein. And one of the things that it cleaves is EAF4G. What does that mean about cellular cap-dependent translation? If you have a protease that's chopping that up, it stops. And so this is a great way, again, to totally over-anthropomorphize, for the virus to have only its RNA be translated, as opposed to all of the cellular RNA being translated. So that cleavage is being used for not only chopping up your polyprotein, but also pushing most of the translation. And when I say most of the translation, why am I saying most of the translation? Because there's also cellular irises and some messenger RNAs. Cellular messenger RNAs also have internal ribosome entry sites, originally discovered with viruses, but then later on found to have this. So what do these viruses look like? There's a whole bunch of different ones, but the main thing here is that they've got lots and lots of secondary structure, and very often in that secondary structure, you have the start codon, which is part of that secondary structure. Really right in there, and a very pyrimidine-rich sequence. Now, what are pyrimidines? Purines and pyrimidines. Purines are big ones. Pyrimidines are the little ones. That's the way I always remember them. What are pyrimidines if you're thinking about RNA? Uracil and cytosine. Man, it's Monday morning, I guess. A lot of people aren't caffeinated enough. Okay, so you're going to have lots and lots of these pyrimidine-rich regions, and as we'll see, particularly cytosine-rich is a very important aspect of not only getting translation to take place, but also for replication as well. There are lots of other kinds of irises. I think there are actually about 6 different kinds of irises. I basically just wanted to point out that there are a lot of them, not that you are going to have to draw the secondary structures of any of these things for exams. But again, this has a pyrimidine-rich sequence. Here the AUG is a little bit further down. But again, it's all these secondary structures which are really important for getting your translation to take place. And I think that our guest lecturer on Wednesday, I'll talk a little bit about this for the flaviviruses as well. But you remember it was a change in the sequence here in the irises, and in fact it's a type 1 iris for polio, where you got attenuation. And so the attenuation so that the virus wouldn't replicate in neurons was due to changes here in this particular sequence. Now, how that directly relates to neurotrophism is still a very open question. We don't even quite understand exactly why that's happening. This vaccine works, and that's all that we really care about. So let's focus in a little bit more on the polyprotein. Again, it's a slightly different figure. This is from the Flint textbook, but basically showing exactly the same thing. What gets released inside the cell when you have infection is this RNA with a protein attached to the end. It's positive strand RNA virus with the iris and a poly A tail. What that means is you just need that RNA in order to get virus replication. So if you can get that RNA inside the cell, you're going to get virus replication. First thing that happens is due to that iris is you have translation. So you end up with this massive, long, again, about 1,000 amino acid protein that again gets chopped up into smaller pieces. And one of the things that's a little different about polio relative to some of the other picoRNA viruses is there are 2 different proteases. There's the 2A protease and the main one, which is the 3C protease, which then chop up this into smaller pieces. First 2A and then 3C. There are a couple of things about this which you should be going, huh, what the heck? How can you have a protease like chopping itself out of a big polyprotein? How's that supposed to work? So it turns out that these proteases are active as part of the polyprotein. And so literally what they're doing is they're cutting themselves out of this protein. So the protein is made by the ribosome, again, over 1,000 amino acids. Those then get chopped up as they're being made by the protein that's coming off of the ribosome, which is still to me really kind of amazing. And if you think about it, here is that cleavage site for the 2A protease. The 2A protease is made right here, right after that. So it's functional as part of this polyprotein. 3C is also true, and this process here, all of this cleavage activity, happens while we have a polyprotein. One other thing to mention here, VPG, which is here at the end of your genome, is made also through proteolysis of this polyprotein here down at the end. So it's all about what? In terms of making these individual proteins. What kind of activity? Scissors, protease activity, right? So that means that everyone is going to do what on our next clicker question? Answer it correctly. So, processing of picornavirus polyproteins, excuse me, let's actually turn this on, is due to the activity of, let's go, come on, there we are. Vote early, vote often. And I'm not going to read all of them. All of the above, yes, exactly.
Probably the worst. I think all the recordings make us up talking too, so everybody else
Now, the only reason to do these is to give me a water break. I'll try to if I get around to it. There were some extenuating circumstances for the last exam. Again, I can see your scores. I know what you voted up here, and I'm just waiting until we get 100%, and I can turn them off. But we don't have 100% yet. I know you're just messing with me. But I can go back and check who's messing with me in this case. Say, one of you insists on messing with me. I don't know what it is. Say, I may have to go and check here and see which one this is. So yes, it's all about the proteases. Okay, so we've talked about how you make the proteins. How do you make genome? This is the same kind of issue as you have with the RNA viruses in E. coli. You've got to deal with transcription versus translation. But here, at least in the case of these picorna viruses, you've got one big polyprotein. If you've stopped translation from that virus, that's it. You don't have multiple other entry sites. And so it's actually a lot easier to deal with. And that's potentially one of the reasons that these viruses have evolved to have big polyproteins. Is there's only one place that you're actually getting a start. And so what happens is once you have enough of the proteins that you've translated, turns out that those proteins are also involved in, not surprisingly, replication, but also in shutting down translation. So there's a secondary structure that's actually 5' of the iris, which is right about here, where you have a binding by the 3CD protein, and that blocks more translation from going on. You also have binding by this poly-C binding protein. What did I keep beating on in terms of iris sequences? They've got pyrimidine-rich sequences. What's one of the pyrimidines? Cytosine. So the poly-C binding protein here, this is a cellular protein. That's what's being shown here. Binds to this secondary structure, and it's a cloverleaf-like structure, but it's not like a tRNA. It's not like we have in all those plant viruses. That's present here at the 5' end of your genome. This blocks translation. Okay, so once you've blocked translation, now what you can do is start replicating from the other end of the genome. How do you start that? One of the things that I didn't mention before, but it's shown here, VPG is actually attached to 2 uracil residues, and it's always attached to 2 uracil residues. So these 2 uracils, what do uracils pair with? A's. So at the 5' end of the genome, you have the complement of what's going to be present at the 3' end of your genome. Turns out that the 3CD protein, when it binds here at the 5' end, will use this sequence here that we're not going to get into in more detail to take VPG, put 2 U residues on it. Then that will get translocated to the other end of the genome. Now we've got UU, and that will get extended. How do those U's get put on here? Well, we have a very convenient amino acid in VPG. And what would that convenient amino acid be? Oops, go forward here. What are convenient amino acids as far as OHs? Tyrosines. So tyrosine residue, U's get attached to that. Again, the OH just like an OH would be in terms of a nucleotide. 2 U's. These 2 U's now will base pair to the 3' end of your genome and replicate all the way down to the other end. This gives us our negative strand, right? Because as always, positive strand, negative strand. The negative strand is here. Then you've got this negative strand, which has a couple of A's at the end, because of course that's what's pairing with these U's. You have more of your VPG with more U's on it. It will start at this end and then make many, many copies in now the same direction that you would be making your translation as well. And so you're never going to have this interaction here. So you don't have to have a nice structure here which blocks translation like you have up here. Now this can replicate on your negative strand and make lots and lots of copies of your positive strand. VPG is now attached covalently because it's got these U residues attached to the tyrosine. At the 5' end, the 3' end had all these U's because it was base pairing with all of these A's, and now you end up with a poly A tail. These guys are now ready to go in order to be packaged into capsid proteins. What makes all of this RNA? It's an RNA-dependent RNA polymerase. This is the 3D polymerase. Again, there are 3 sections of your polyprotein, each of that get cut into smaller sections. 3C is the main protease. 3D is the polymerase. The structure of this polymerase was, I think, the very first structure of an RNA-dependent RNA polymerase to be made. And what was the big surprise? It looks like a DNA polymerase because it's got a thumb, fingers, palm. In the palm structure is where you have your active site. So these are DNA polymerase-like, and so actually the DNA polymerase structure is probably like an RNA-dependent RNA polymerase because authors came up with DNA in the first place that, again, we can argue more about later as you like. But these structures, the RNA-dependent, RNA polymerases have these right-hand structures. What else have we talked about that has this kind of right-hand structure? Single subunit what? T7, RNA polymerase, which is a DNA-dependent RNA polymerase, has basically the identical structure to this. So an RNA polymerase is dependent on DNA, and an RNA polymerase is dependent on RNA. And as we'll see much later, reverse transcriptase also has basically the identical structure to this. So the primordial polymerase structure, nucleotide polymerase structure, is probably this kind of structure. So we've made our genome. We need to package it inside a virion. How does that get done? Well, again, we've got our precursor protein, which gets chopped up into smaller pieces by our 3C protease. These guys get assembled into trimers, which are each of the faces of our icosahedron. Those become pentamers, and these pentamers get put together, interestingly enough, in the absence of any kind of scaffold. So this is quite different than what we've talked about for a lot of the bacterial ones. In the absence of a scaffold, these get put together with the RNA, packaged around the RNA, and then this is released from the cell. What's important here is that this assembly process is not taking place between VP1, 2, and 3. It's VP0, VP3, and VP4. What the heck is VP0? VP0 is still a precursor which only gets cleaved when you have your final structure here. So VP2 and VP4 are one protein, and you get assembled as these two together, and only after you have complete assembly do you have the proteolysis here. Now, this particle is infectious. This particle with VP0 is not infectious. And so it's a way you can get the unidirectionality, getting your metastable state of all of these virions, is by having this proteolytic cleavage that happens right here at the very end of the actual assembly process. We already talked about how you shut down host cell translation by chopping up EIF4G. What does EIF4G do? Binds to the cap, which is a bridge from the cap. EIF4E is what binds to the cap. Turns out that the 3C protease also inhibits the cellular DNA-dependent RNA polymerase, also inhibits vesicle transports. One of the things that I forgot to mention about the replication process, you may have noticed, I'm going to back up here really quickly, here, is that the VPG protein is part of this 3AD protein, which binds to membrane vesicles. And we talked about membrane vesicles in terms of those plant viruses replicating to escape from the RNA interference machinery. Here we have replication at vesicles, probably just by pulling all of the appropriate proteins together. And if you're dependent on vesicles for replication, it's going to be to the advantage of the virus to have lots and lots of vesicles. And so the viral proteins also block vesicle transport, so they get stuck inside the cytoplasm. And through all of these processes, what you can see, if you just look at all of the proteins that are being made by a cell, and the way you do that is just take your cell, label it with radioactive amino acids, and look, you'll have all of these different proteins. You can't resolve them relative to each other. And you look at all the proteins that are being produced over time after an infection process. You'll notice that almost all of the proteins here are your viral proteins that are being made. So it's an incredibly efficient process once you get this infection that has taken place. Okay, well, a couple of little vignettes to finish up here. The first one is, we mentioned in one of the very first lectures, polioviruses 3C, 332,652, etc. It's completely molecularly defined and was made by the Wimmer group, Eckhard Wimmer. I think he's at Stony Brook, if I remember correctly. And there was a huge discussion about whether this was good research to do, is making an infectious virus that has killed literally millions of people worldwide. What's going on there? I think it's really kind of overblown. And the reason for that is basically this. So the original publication where the Wimmer group, from one of the leading groups working on poliovirology, showed that you can make an RNA and take that RNA and put it into cells, and with that RNA make poliovirus, so basically purify the RNA from poliovirus, do a transfection, take that RNA just by itself, put it into cells, you make poliovirus. It took that lab 11 years to come up with a synthetic DNA that they could use to make polio with. Part of that process, however, is they ordered all of this DNA. The poliovirus genome is pretty small. It's only about 4,000 nucleotides. And getting all the DNA to do that, you have to buy it from people. And they bought this from a whole bunch of different suppliers, which at that point would make it for them. Now if you try and order a piece of DNA which encodes a virus genome, all kinds of red lights flash and your synthesizer stops working. And so that is also part of the process, and potentially another reason that we do need to still keep vaccinating. Is that this is something which is still quite doable and makeable. So whether we'll be able to stop immunizing completely for polio, I think is a pretty open question. Rhinoviruses, these are actually, I think, way more important than polio, particularly since we're about to eliminate it. Very similar, couple of different things. This is a few years ago now. One of the great things we have now is a lot of sequencing. All of these rhinovirus genomes have iris sequences. All of them have 5' cloverleaf-like structures, so probably they're replicating in the same way. Interestingly enough, they don't all have 3' poly A tails. Many of them have hairpin-like structures, and probably that's protecting the genome a lot like what you see with the tRNA-like structures that we have in plant viruses. And this was written up a couple of years ago now. I mentioned enterovirus D68. 2 years ago, almost 1,200 cases, most of them in California. A few cases of acute flaccid paralysis. Whether that had anything to do with this virus infection, and these are the virions. You get very high numbers here, and I love this one because each of these black dots actually represents a virion. So you have huge numbers of virions being produced here. Seems to be causing a very mild respiratory illness. It's an enterovirus. Why is it calling it a respiratory illness? Again, that's part of the problem with naming. These were originally found in stool samples, but probably are being transmitted through aerosols. Whether it has anything to do with paralysis, who knows? But these are the kinds of things which are keeping popping up again and again. Yeah, Trevor?
What tissue is that right there? That it's
Oh, this particular one? I don't know. I go and look at the link down here, and it would tell me. So that's all for polioviruses. Sorry, picornaviruses, I should say. They're very common. They're all over the place. We've known about poliovirus disease for over 5,000 years. Almost gone, at least polio, and I think probably wild polio certainly in the next couple of years, and hopefully even vaccine-derived in most of our lifetimes. The important stuff, of course, is all this stuff after that. CO3 equals 3 structures, internal ribosome entry sites, proteases, the VPG protein, which is the primer for your synthesis, also really critical for getting the genome inside the cell. Those all come together. How do you make your host unhappy? By cleaving up the translation proteins. And then we had some more of the disease processes. If you're interested in more about how to eliminate polio and what people are doing, this is a really great website where they get into a lot of the details. And in fact, most of the data that I showed at the beginning in terms of the progression of eliminating polio all comes from this website. So, guest lecture on website, flaviviruses. What's our favorite flavivirus? Zika.